everyone and welcome to Miss Electric Biology. In this video we're going through all of OCR A Module 6. Now I know your time is precious because you're revising for your exam, so if you do want to skip ahead then I put the time codes at the bottom so you can pick and choose which parts of this chapter you most need help with. And speaking of help, if you do want that extra push and support in creating notes and knowing the key marking points, then definitely check out my OCR A notes or my OCR A flashcards, which cover all the key marking points to help you to remember what to say in an exam question. And I'll link them both below. But for now, let's get into it. So in topic 6, these are the different parts that we're going to be going through, starting with cellular control. And we're looking at gene mutations. A gene mutation is a change in the base sequence of DNA. And they occur randomly during DNA replication, which is within S phase of interphase. These random mutations are more likely to occur if you're exposed to mutagenic agents, and these are different things that increase the likelihood or the rate of mutations happening. And that includes high energy radiation, such as UV light, ionizing radiation, such as gamma rays and X-rays, and then chemicals, which would be known as carcinogens, such as mustard gas, and a whole range that are in cigarette smoke. The two types of gene mutation you need to know are deletion and substitution. So if we have a look at an example, here is our original DNA base sequence, and a substitution is when one of those nucleotides, and therefore bases, is swapped for a different base. And that now means we've got a different triplet of bases, and it might code for a different amino acid. Sometimes, though, it will still code for the same amino acid because the genetic code is degenerate, meaning multiple codons can code for the same amino acid. So that would be known as a silent mutation because the overall polypeptides chain would have the same amino acid sequence. A deletion mutation is when one of the nucleotides, and therefore bases in the sequence, is removed. And we can see that one of these 3 cytosine bases has been removed. And as a result, we get what's called a frame shift. And that is when all of the bases will move one position. And in this case, they'll all move one to the left because a base was deleted. And that then means all of the subsequent codons will code for a different amino acid. They've all been changed, and that will have a huge impact on the overall protein.
For this OCR 2025 version, I've also inserted, which is relevant for what I'm inserting in the information about a insertion mutation. So you do also have to know about the effects of insertion mutations, which is when one extra base is added, and that also causes a frame shift. It shifts all of the bases one position to the right. So let's take a look then at the effects of these gene mutations. Because mutations alter the gene, they can result in a different amino acid sequence or the primary structure in the encoded polypeptide. If the amino acid sequence changes, then this has an impact on the folding and the coiling of the secondary structure, and therefore it will be different. When the protein is modified into the tertiary structure, it will form the hydrogen and ionic bonds in different places, and that causes it to fold differently. And therefore, it results in a different 3D shape, and therefore it has a different function, which is why it could become a non-functioning protein. A base substitution could be silent, though, which means that even though the base was swapped, it was substituted, it might still code for the same amino acid, as in the codon might still code for the same amino acid. And this is because the genetic code is degenerate, which means multiple codons code for the same amino acid. Base deletions and insertions, collectively known as indel mutations, result in frame shifts. The removal or insertion of one base changes all of the subsequent codons after the point of the mutation. It says, as can be seen above, this was actually on the previous slide. This is more harmful as multiple amino acids may be incorrectly coded for, and therefore have a bigger impact on the change of shape of the final protein. Not all mutations are harmful though. Sometimes a new protein that is made might be beneficial, such as the mutation that resulted in antibiotic resistance in bacteria. And some effects are neutral, so if they do not code for a protein that alters the survival chances of that organism.
Now, protein synthesis can be controlled, and one way it can be controlled is through transcription factors. And transcription factors are molecules that can bind to the DNA and either initiate or inhibit transcription. These molecules will move from the cytoplasm into the nucleus and they bind to the DNA. Transcriptional factors are proteins, so the reason they can bind is they'll have a unique 3D shape which will be complementary in shape to bind to one particular sequence of bases and therefore one particular gene. When they bind, it enables RNA polymerase to bind, and in that way it will initiate transcription so that mRNA can be created, and then that can go to be used in translation. If you don't have the binding of a transcriptional factor, the gene is inactive because transcription cannot occur. But as I said, sometimes the transcriptional factors are ones that actually inhibit RNA polymerase from binding, and therefore it will prevent transcription. But in both of these ways, transcriptional factors are controlling this DNA being copied into mRNA, and therefore they're controlling gene expression. So here's a quick summary of what we just went through there. The fact that transcriptional factors go from the cytoplasm to the nucleus, they bind to the DNA to either turn on or off a gene, but the mark scheme tends to want you to not use that phrase and instead say that it can either initiate or inhibit transcription, and in that way it's controlling protein synthesis. In prokaryotes, operons are involved in controlling transcription, and an operon is a group of simultaneously controlled genes that are either all expressed or not. And this is more common in prokaryotes than in eukaryotes. And we're going to focus on the lac operon, which is found in E. coli, and it's a sequence of 3 genes, which we can see here, lac Z, Y, and A. And this set of genes are collectively involved in lactose digestion. Bacteria require less energy to break down glucose than lactose. So glucose is the favourable respiratory substrate. However, if glucose isn't present, then lactose will be digested and used. So therefore, proteins produced by the lac operon are only needed if glucose is absent and lactose is present and therefore need to be digested. So this regulates the transcription of these genes to meet the demand. So the 3 genes that we saw in the previous image, lac Z, Y, and A, are in this operon. And the lac L is a regulatory gene found near the start of the operon. So I'm just going to go back to show you that. Here's our lac L, and that is at the start, near the operon. And that gene codes for a repressor protein that inhibits transcription when there is no lactose present. So when the lactose is absent, that repressor protein is constantly produced, and it binds to the operator, which is a region close to the operon, which prevents RNA polymerase from binding to DNA at the promoter region. And in this way, transcription is inhibited. If lactose is present, the lactose binds to the repressor protein, and this causes it to change shape. This change in shape prevents the repressor protein from binding to the operator, and therefore RNA polymerase can bind to the promoter, and transcription of the lac operons occurs. Cyclic AMP also has a role because it increases the rate of transcription of the lac operon. In order for sufficient enzymes to be produced by lac Z, lac Y, and lac A, cyclic AMP receptor protein, CRP, must bind. And the CRP can only bind and increase the rate of transcription once it has bound to cyclic AMP.
Next then, let's have a look at the post-transcriptional changes. So mRNA gets modified once it has been transcribed. So this newly synthesized strand of mRNA is called pre-mRNA or primary mRNA. And then once it's modified, it's known as mRNA. The key modifications are the removal of the introns and the rearrangement and reordering of the exons. And that's what we can see here. The introns are removed and those exons would be rearranged and reordered. All of the introns need to be removed as these do not code for amino acids in the primary structure of the polypeptide being made. These are removed by a protein called a spliceosome, and therefore the removal of them is known as splicing. The exons can be reordered and some are removed to allow a single gene to actually result in the creation of multiple proteins, and this process is called alternative splicing. There are also changes that happened after translation, so post-translational changes. In addition to the activation of proteins by CAMP, as discussed linked to the lac operon, proteins are modified further after translation. Non-protein groups can be added to the protein. So, for example, lipids and carbohydrates, that could make a glycoprotein. This happens in the Golgi apparatus. The proteins are then folded into their unique 3D shape and bonds are formed to hold that 3D shape in place. Some proteins are made in active forms and they need to be activated by phosphorylation.
We then move on to the genetic control of body development, and this is where we then look at the homeobox gene sequence and Hox genes. So plant and animal cells and fungi all have homeobox gene sequences to control the development of their body. Homeobox genes are sequences of genes which create proteins that regulate the expression of other genes that are involved in the formation of the body in the early stages of an embryo. These homeobox gene sequences are highly conserved, which means they're very similar in plants, animals, and fungi. And that's what we can see here in this pretty complicated diagram. All of these different genes and the sequences of them, we can see the colour, and that links to the image, the part of the body development it's involved in. And we can see that it's pretty similar between all of these very different organisms. So a bit more about Hox genes. These are a type of homeobox genes found in animals, and they're responsible for the correct body development and body positioning of the different body parts. The order of these genes in the DNA is the order in which their effects are expressed on the organisms. And many animal bodies are segmented. So, for example, an insect has 3 segments, and there are segments on a worm, that's what all the rings are, and invertebrates, such as humans, all of the vertebrae in our backbone are segments. The body development of most organisms also shows symmetry. Some have radial symmetry, like a jellyfish, meaning the top and the bottom are the same. Others have bilateral symmetry, like humans, meaning the left and the right-hand side has symmetry. We then look at mitosis and apoptosis because these both also play an important role in the development of body forms. And that's because mitosis results in an increase in the number of cells, which results in body growth. And apoptosis, which is programmed cell death, will remove unwanted cells. The cell cycle is controlled by genes to ensure new cells are only made when you need growth and repair to occur. And that's to preserve energy, but also to prevent tumours from forming. And there are 2 genes that help this. The tumour suppressor gene, which is responsible for making proteins that stop the cell cycle from occurring if you don't need any more cells, and proto-oncogenes, which are responsible for producing a protein that initiates the cell cycle when you do need new cells. If an error is detected in a cell, or if it's too old to function, that's when apoptosis occurs and the cell is destroyed and the useful resources get recycled. So the control of mitosis and apoptosis are both in response to internal and external stimuli such as stress. Hox genes also regulate mitosis and apoptosis. The genes that regulate the cell cycle and apoptosis are impacted by external and internal stimuli, so for example temperature on the outside, and hormones and psychological stress in terms of internal stimuli. And the effect of this is highest during growth development. The next part of this topic is inheritance. And we start by looking at this whole range of key terms and their definitions. So I suggest you pause at this point and turn this into a set of flashcards, and you can use this to constantly test yourself to check you know these definitions that could come up as 1 or 2 mark questions.
This is an edited in extra point for the 2025 version. I want to draw your attention to the fact that I have changed the definition for codominant here to match exactly what is wanted on the mark scheme. Both alleles are expressed in the heterozygote, resulting in a different phenotype. So make sure that is the definition that you are using.
You also need to be able to do genetic diagrams to be able to determine potential phenotypes in offspring. So these are the different coding systems that you need, and we'll see how these are applied in a range of examples. As a brief overview, monohybrid is when there is just one gene and you either have dominant or recessive, shown by a capital or a lowercase letter for that particular letter of the alphabet. Codominance is when you have 2 genes that are both dominant and both expressed. So we can't use that coding because they both need a capital letter. So instead you use 2 different letters, which would both be capitals to show their dominance, and the base letter represents the gene. Multiple alleles, we do the same coding because multiple alleles is when you have more than 2 alleles for a gene. So we can't just use one letter, capital and lowercase, because we need more than 2 codes. So again, you choose a base letter to represent the gene and superscript to represent the allele. For sex linkage, the base letter that is used is to show whether it's an X or a Y chromosome. And then in superscript, you would show the allele. Autosomal linkage and epistasis are both involved in dihybrid inheritance. So that's why we have 2 different genes shown and they're represented by different letters. But in both cases, it's just the capital letter is the dominant, the lowercase is the recessive. So let's have a look at this in action, starting with monohybrid. And for all of these examples, I suggest pause it, have a go yourself, and then mark it as I go through. So we're told that cystic fibrosis is caused by a recessive allele. And the question is, if 2 carriers reproduce, what's the probability they'll have a child with cystic fibrosis? And what's the probability they'll have a girl with cystic fibrosis? So step 1 is always working out the parental genotypes. And if they're both carriers, that means they don't have the condition, so they must have the dominant allele, but they carry the recessive allele. Then when we do our Punnett square, we need to show the gametes, so the alleles that are in those gametes for both parents. Then we can show if those gametes fuse, what are all the possible genotypes, and then it's good practice to put in the phenotype into each box to show what that genotype would result in. So these 3 which aren't labelled would be someone that doesn't have cystic fibrosis, but we can see here there's 25% probability that they'll have a child with cystic fibrosis. So the probability they'll have a girl with cystic fibrosis is half of that, because there's always 50% probability you'd have a girl or a boy. So that's why the second part would be 12.5%. Then if we move on to a codominant example, we're told that cows can be red, white or roan in colour, and red and white are both dominant. If 2 roan cows reproduce, what is the probability they'll produce red cows? So this is what roan looks like, where you have both alleles are expressed, the red and the white, even though the red is kind of more brown in colour, we still call it red. So if both parents are roan, that means they have the red and the white allele. So this is the genotype for both parents. Therefore, these are the alleles in the gametes for both parents. And when we look at those fusing together, we can see the 4 possible genotypes. And I've matched that up to the phenotypes. And from that, we can see there's 25% probability that they would have a red offspring. The next example is multiple alleles. And this often goes hand in hand with codominance. So we're going to have a look at the example of blood group. There are 4 possible blood groups, and these are all the genotypes that would code for those blood groups. So blood group A means they've got an A-shaped protein, B, they've got a B-shaped protein on the outside of the red blood cells, AB means they have both, and blood group O doesn't have any protein on the outside of their red blood cell. A and B are both dominant, O is recessive. So the information that we're told then is, you've got parents with blood group AB, and the other parent is blood group O, and they reproduce. IA and IB are codominant and IO is recessive. And we need to say, what is the probability they'll have offspring with blood group O? So step 1, we need to look at the parental genotypes, and that is for AB, they'd have one of both of the dominant alleles. Blood group O is recessive, so they'd have 2 copies of blood group O. Then we do our Punnett square to show what the gametes would contain. Then we can fuse all of those gametes together to show the genotypes, and then I've added in the phenotypes. And we can see that the genotypes along the bottom both have an A and then the recessive O. So we have 50% probability of blood group A. So for both of those, it'd be blood group A, and therefore 50% probability of blood group A. Next then, a sex linkage example, and we're told that colour blindness is caused by a recessive allele found only in the X chromosome. And we've got a parent who is a non-colourblind male who reproduces with a female carrier of the allele. And we've got to say, what is the probability that their children will be colourblind? So step 1, we need to work out the genotypes of the parents. So the mother would be XX, because that codes for biologically female. And because they are a carrier, they have the dominant allele, meaning they don't have the condition, and they carry the recessive. The male would be XY, so biologically that's XY, and the X capital R is because they do not have colour blindness. And there is no allele on the Y chromosome in sex-linked examples because it's too small to carry as many of the genes as the X does. So then we see the potential gametes, the genotypes and the offspring of those gametes fuse, and then I've written in the phenotypes. And we can see that the probability that they'll have a colourblind child is 25%. Now, usually you should actually add in the sex as well. So this would be a female with normal vision, a female with normal vision, a male with normal vision, and a male who is colourblind. We then move on to epistasis, and this is when one gene influences the expression of another. So that means one gene can either affect or mask the expression. And three common examples are coat colour in mice, coat colour in dogs, such as Labradors, and fruit colour of vegetables. So we're going to go through an example with the coat colour in Labradors, and the two genes are gene E, which is controlling whether pigment is expressed or not. And the dominant E will code for pigment being produced. The recessive allele, if an individual has two copies of that, that means they won't make any pigment, and the dog therefore will always be this yellow colour. The second gene codes for what colour pigment would be expressed if they have the dominant E. So capital B would code for black fur, lowercase b would code for brown fur, but you would only get black or brown if, for gene 1, they have a capital E. Because as soon as they have two recessive E's, that means it doesn't matter what alleles you have for the second gene, they will always be yellow. And in that way, it's epistasis. So here's an example that we've got. This time we're giving the parents genotypes, and you have to work out the phenotypes. So we can see that both parents have a capital E, which means pigment will be expressed. They both have a capital B, which means they're both black. Now, because this is dihybrid inheritance, we now need to work out all the possible gametes that these two parents could produce. And that would be capital E, capital B, capital E, lowercase b, lowercase e, capital B, lowercase e, lowercase b. So for both of those dogs, those are the four possible gametes. So we put that into our Punnett square. Then we can see all the possible genotypes from those gametes fusing, and then I've written in the phenotypes for all of those. And we have a ratio of 9 black, 4 yellow, and 3 brown. So this leads us into the idea of dihybrid crosses, and these are crosses when you're looking at two genes being inherited at the same time. And a common example is Mendel and his peas. And the two genes are one that codes for the shape of the pea and one that codes for the colour. So here we've got a round and yellow pea and a wrinkled and green pea. So whenever you do these genetic crosses, just as we've seen in the ones we've done so far, you always need to make sure you're showing the parental phenotype, parental genotype, possible gametes, the offspring genotype, the offspring phenotype, and the proportion. Those are typically your marking points. So for this example, our round yellow, in this case, they would have to tell you that it's homozygous dominant, because otherwise that could be heterozygous for both. So they'd have to let you know that. But for wrinkled green, wrinkled is recessive, green is recessive, so it would be homozygous recessive. But you aren't expected to know that the round is dominant and the wrinkled is recessive. They would have to give you that information. So then if we look at the possible gametes, this one can only produce capital R, capital Y, and this one can only produce lowercase r, lowercase y. So when you work out the gametes in a dihybrid cross, each gamete should have one of each letter. So then, because this one has only dominant and this one has only recessive, that would mean all of the gametes would have to have one of each. They'd be heterozygous for both genes. And in this case, that would mean all of them would be round and yellow. So that's quite a simple dihybrid example. But if we then cross that F1, which means the first generation, which was heterozygous for both genes, it now gets more complex because it's not so straightforward in terms of the gametes. We have to work out all of the possible combinations of the R and Y we could get in the gametes, and these are your 4 possible combinations for both parents. Then when we get to our Punnett square, it's going to be 4 by 4, because there were 4 gametes that each parent could produce. Then if we fuse all of those together, we get the genotypes. Then we fill in what all of the possible phenotypes are, and we can see we've got this 9 to 3 to 3 to 1 ratio. And in a dihybrid cross, if both parents are heterozygous for both genes, you will always get this ratio, 9:3:3:1, unless there is autosomal linkage or crossing over or epistasis. So that's not written down there, but also epistasis would affect that ratio. This takes us into then what autosomal linkage and crossing over are and how they affect that ratio. So recap on crossing over, that is when in meiosis you have the non-sister chromatids of a homologous pair overlapping, chromatids break, and you swap sections of the chromatids. And as a result, we've now got a new combination of alleles in the gametes. And that then is going to change the gametes that you write in your Punnett square, and therefore your overall prediction. Autosomal linkage is when the 2 genes that you're considering in this dihybrid cross are both on the same chromosome. So if we were thinking about gene 1 for shape and gene 2 for colour, we're saying they're both on the same chromosome. So if we stick to the example we were just doing with 2 heterozygous parents, which meant they had a dominant R and a recessive R, a dominant Y and a recessive Y, we're shown here that they are linked on the same chromosome. And in this particular example, the 2 dominant alleles are on one chromosome and the 2 recessive alleles are on the other chromosome of the homologous pair. So that means it's not possible to get the 4 gametes that we'd written. It's only possible to get a gamete which would contain this chromosome, which would mean capital R, capital Y, or a gamete which would contain this chromosome, which would be lowercase r, lowercase y. So because of this autosomal linkage, we've reduced the number of different types of gametes we could get, and there's now actually only 2. So instead of there being the 4 possible options that we said when there was no linkage, we now only have 2 options for both of the parents. So now if we do our Punnett square again and look at all the possible genotypes of the offspring, the phenotypes this time we have 3 yellow to 1 wrinkled, and we haven't got that 9:3:3:1 ratio because those genes were linked onto one chromosome, and therefore it reduced the number of possible gametes that could be made. So this is just summarising in this example. In dihybrid, we would have got a 9:3:3:1 ratio, but with the autosomal linkage, we got a 3:1. So the way that this often comes up in exam questions is they would say, so the way this links to crossing over is from that autosomal linkage, we said we expected a 3:1 ratio. In an exam question, they might say though, however, this is what the scientists observed. They actually saw 4 phenotypes, not just yellow and round and green and wrinkled. They saw all 4, and these were the numbers they observed. And this would only be possible if crossing over happened to create new combinations of alleles in those gametes. So let's just have a look at what we mean by that. In crossing over, we've got our homologous pairs. The non-sister chromatids in a homologous pair can tangle up, break and swap those alleles. So now, instead of just having the lowercase r, lowercase y, capital R, capital Y, because those 2 parts have swapped, we now have new combinations of alleles in the gametes. We have the original lowercase r, lowercase y, capital R, capital Y, but we now have these recombinant examples as well, where we've got the capital R and the lowercase y, and the lowercase r and the capital Y. So in terms of how we'd explain that in our answer, the yellow and round and the green and wrinkled are most common because they're resulting from two gametes fusing together that weren't as a result of crossing over. It was the original two possible gametes. That's why they're the most common, because crossing over is actually quite rare. It doesn't happen all the time. Then the reason we have fewer green and wrinkled compared to yellow and round is green and wrinkled are both the recessive alleles. So then explain why we got hardly any of the yellow wrinkled and the green round. That is because crossing over is rare. So we won't get very many of the capital R lowercase y or the lowercase r capital Y gametes. And those are needed to create the genotypes that would code for those phenotypes. So that is why we don't have very many of those two phenotypes. Now we can actually link inheritance to a statistic, and that is chi-squared. And that's because we're counting how many individuals fit into a category, which would be a particular phenotype. So let's look at how you could use chi-squared in inheritance. So in this example, we're going back to the cystic fibrosis example that we did earlier on, where we said there was 12.5% probability of having a girl with cystic fibrosis. We can actually use the statistic chi-squared to investigate whether what we expect, 12.5%, is significantly different from what you actually observe in reality. So do we actually have a significant difference or not? And we'd be hoping that we'd find there is no significant difference between what you expect and you observe, because that would tell you that your prediction in terms of your Punnett squares are accurate. They are showing what happens in inheritance. So we're going to do an example with corn. Corn can actually be purple or yellow in colour, or smooth and wrinkled in texture. So if we use this example where we've got dominant purple and recessive yellow, here are our possible gametes, our possible genotypes and phenotypes, and we're showing that we are expecting a ratio of 3 purple to 1 yellow if we were to breed those two parents plants of corn. And we're then told a student counted the actual number that those two parents produced, and there were 21 purple kernels and 13 yellow kernels. And you then need to say, does what we observed match what we expected from our Punnett square? And this is where we use chi-squared. So our null hypothesis would be, there is no significant difference between the expected ratios from our Punnett square and the observed frequency of what we actually saw in those corn kernels. So here is our table to work out the chi-squared. We said that we observed 21 purple, 13 yellow. So that means in total there are 34 corn kernels. Now we expected a ratio of 3 to 1. So we then need to put that as an actual frequency. So what is 3 parts of 34 and 1 part of 34? It's 25.5 and 8.5. We then do observed minus expected. We then square that, divide that by the expected to give us our overall chi-squared value of 3.176. In this example, we have 1 degree of freedom because we have 2 categories and it's always number of categories minus 1. So the next step is comparing your calculated value to the critical value for 0.05. So for us, we said we had 1 degree of freedom. So that critical value is 3.841. And our calculated value of 3.176 is lower than that critical value at p equals 0.05. So what that means is there is more than 5% probability that the difference between our expected and observed is due to chance. So we have to accept our null hypothesis, which was there's no significant difference between what we observed and expected. And that is what we want. That means the corn kernels did follow the expected ratio of 3 to 1, and therefore it followed the Mendelian genetics that we were expecting. Now, something else that you can use is the Hardy-Weinberg principle, and this is a mathematical model that's used to predict allele frequencies within a population. So just a couple of key terms to help with your understanding of this, which again, pause, turn it into a set of flashcards. Gene pool, population and allele frequency, here's what it means. And we need this idea for, in particular, the allele frequency to know this formula. So the Hardy-Weinberg equation, there's actually 2 formula to use here. p squared plus 2pq plus q squared equals 1. P plus q equals 1. Now what this actually means is the p plus q equals 1. P is telling you the frequency of the dominant allele. Q is telling you the frequency of the recessive allele for one gene in a population. And that always has to equal 1, because if this is just for one gene where we just have two possible alleles, if you add up both of those alleles, that will be the total. That will be all of them in the population. The top equation is the same idea but for the genotypes. So p squared is homozygous dominant, 2 copies of the dominant allele. Q squared is 2 copies of the recessive allele. And 2pq is when you are heterozygous, you have 1 of each allele. So I'm going to keep that at the top and go through an example with you. Cystic fibrosis is caused by a recessive allele. And we're told that 0.02% of the UK population suffer from cystic fibrosis. And you have to say what proportion of the UK are carriers. So the first step is always working out which bit of the formula have they given you and what are you trying to work out. So if 0.02% of the population suffer from cystic fibrosis and that is caused by a recessive allele, that means cystic fibrosis is homozygous recessive. And that was q squared. So q squared equals 0.02%, but we always have to convert this back into the decimal, so divide that by 100. Now they have said what proportion are carriers. The carriers are heterozygous. We want to work out 2pq. So to work out 2pq, we need to know what p is and what q is. So if we know q squared is 0.002. If we square root that, we now have what q is. We can then use this formula, p plus q equals 1, to work out p. So we'll rearrange that, and it'd be 1 minus q, and that tells us that p is 0.986. Now we just said that we're working out 2pq, because that is the carriers. And 2pq then, we just do 2 times p times q. And that gives us 0.03. Now, if they don't tell you whether they want it as a percentage or not, in the mark scheme, they'd accept either of those. But obviously, if they say give us a percentage, you'll have to times that by 100. Then we get to the idea of continuous and discontinuous variation. And this means that the variation within species can either be classed as continuous or discontinuous. Continuous is when it's a type of variation where there is a whole range of values, for example, height or mass, whereas discontinuous is categorical, for example, blood group. You fit into a particular group. So data representing continuous variation would be presented with a histogram, where discontinuous would be presented with a bar chart. And this here is just showing you a summary of the differences between these two types of variation. So again, this would be another great example to turn this table into a flashcard where you have continuous variation on one side, the different properties on the other, and then another flashcard for your discontinuous variation. Then we move on to types of selection, and this links to the idea of natural selection. So when natural selection occurs, there are 3 possible outcomes. We could have stabilizing, directional, or directional selection is when only one of the extreme traits has the selective advantage. And that's usually when there's a change in the environment or a new predator, a new disease. So those individuals with the extreme trait are more likely to survive, and the other individuals are less likely to survive, and therefore they'll die out. So we see over many generations a change in what the mode is, and an example of that is antibiotic resistance in bacteria. Stabilizing selection is when the modal trait remains to have the selective advantage. So there's no change in what is the modal value, but more individuals will have that modal trait. The individuals with the extremes will start to die out, so we'll lose those extreme traits. So the range in alleles becomes narrower, and the mean, even more individuals will have that mean. So that means the standard deviation would decrease. And an example of this is human birth weights. Now, linking back to this idea of speciation that we talked about with the disruptive selection, speciation is the creation of a new species. And this happens when one original population becomes separated so that they are reproductively isolated, which means those two isolated populations can no longer breed together. This can result in the accumulation of differences in these separated populations in their gene pools to the extent that those two populations become so genetically different over many, many years of accumulating different mutations that if they were to then reproduce, they wouldn't be able to make fertile offspring because they're now too genetically different. And that's why they get classed as two different species. Now, what causes a population to become reproductively isolated could be some sort of geographical barrier, and that's known as allopatric speciation, or they might be in the same location, but there's something that changes about their reproduction mechanism, so they don't reproduce together, even though they physically could reach each other, and that is sympatric speciation. So let's just go through those two. Allopatric is when they are separated geographically. So it could be maybe there's a new mountain range following an earthquake, and that population becomes geographically isolated. That separates the original population into two, and now they are unable to reproduce. Both separate populations will accumulate different beneficial mutations over time through natural selection. And due to this accumulation of those different DNA sequences, they'll become so genetically different that they aren't able to reproduce and make fertile offspring, and that's why we class them now as two new species. Sympatric speciation is when they are not geographically isolated, so they are in the same location, but something changes in the way they reproduce or their behaviours around reproduction, so they just won't reproduce together. So this could be because of a random mutation in the gene that codes for their courtship ritual, and therefore maybe individuals don't recognise their courtship ritual and they don't reproduce with them. Or it could be a mutation that results in them producing eggs or whatever the gamete is at a different time of year. In terms of plants, it might be that they start flowering at different times of year, so they can't reproduce with the other plants that flower at a different time. So due to this, again, you have this reproductive isolation. They accumulate different mutations. There's no gene flow because they're not reproducing together. And over time, they become so genetically different, they can't produce fertile offspring, and they're classed as two different species. Genetic drift is the idea that in every generation, there is a change in the allele frequency because you are not a clone of your parents. There will always be genetic drift from one generation to the next, and it's only when there is a large change in the allele frequency, so substantial genetic drift, that we class it as evolution. But genetic drift has a bigger impact on smaller populations because it's all to do with frequencies. So if you've got a smaller population, even a small number changing will be a bigger percentage compared to if it was a large population. So that's why genetic drift has bigger impacts on smaller populations. Genetic bottlenecks and the founder effect are the next ideas here, and we're going to look at genetic bottlenecks first. These are caused by events that kill almost all of the population, leaving only a few individuals left behind. And this has happened in the past because of overhunting of some animals. So here is the original population size. There might have been overhunting, so now the population size has decreased, but maybe the ones that were left behind didn't actually have all of the alleles that were represented in the original population. So we've decreased the gene pool. So many alleles for those genes are lost, and the remaining breeding populations will keep passing on those same limited alleles. So it results in a lack of genetic diversity, and therefore genetic diseases that exist in the population are far more likely to be passed on. The founder effect is very similar, but the reason it happens is different. The founder effect is when a few individuals from an existing population relocate themselves to a new isolated area. So we can see here we've got an original population. A small number of them migrate to this isolated area. And we can see that this gene pool is not the same as this one. In the original, there were more purples than reds. Now we've got more reds than purple. But over time, This is then going to reduce the gene pool because those individuals will keep reproducing together and passing on the same alleles. And we can see here in this example, we've got now what was a mixed set of alleles, we've only got one particular allele. So this idea of genetic variation can occur through natural selection, but also artificial selection has an effect. This again would be yet another good set of flashcards. So natural selection is when individuals within a population show a range of phenotypes because there is genetic variation through mutations. Now, environmental factors, or it could be biotic factors like predators and disease, will determine which individuals are more likely to survive. The ones that survive will pass on their alleles to the next generation, and then that allele will become more common in the population. And in that way, that allele is naturally selected for over many generations. Artificial selection is when humans deliberately select which plants or animals reproduce together based on the characteristics they want to see in the offspring. So humans here are artificially manipulating the gene pool so that the favourable alleles become common and the less favourable alleles become less common. This was particularly prevalent in creating dog breeds with popular features, for example, pugs. Pugs were deliberately bred by humans because it was deemed cute to have this flat, squashed up little squishy face. However, there's lots of ethical issues around this because it didn't allow natural selection to occur, which selects for the alleles that increase the survival of the species. And because humans selected characteristics based on what they thought looked cute, it's actually had a negative impact on their survival. Pugs, because they have this squashed face, they don't have as much space in their nose for breathing, and they often have difficulties breathing, and it can result in lots of medical issues. We then get to gene banks, and an example of this is seed banks. So seed banks are where seeds are stored for lots of different plant species, so that you've got a bank of that DNA in the seed in case they went extinct. These are also a way of maintaining genetic material for use in selective breeding. So as we just said, gene banks, they're stores of biological samples, for example, plant seeds, or it could be animal semen or eggs if we're thinking about animals. Selective breeding often involves inbreeding, so we use gene banks to help increase genetic diversity by deliberately breeding together the seeds or the gametes of animals from individuals that we know are not closely related. So this is important as it reduces the frequency of homozygous recessive diseases in offspring. Next, we get on to manipulating genomes. We start by looking at DNA sequencing techniques and in particular sequencing projects. So we need to know the fact that the genome of an organism is all the genetic material that it contains. And sequencing projects have read the genomes of a wide range of organisms like humans. And this provides opportunities to screen DNA to identify potential medical problems. The human genome consists of 3 billion base pairs and approximately 20 to 23,000 genes. The proteome is all the proteins that a cell is able to produce. With simpler organisms, if the genome sequence is known, then the proteome can be derived from the genetic code. This may have many applications, including the identification of potential antigens for use in vaccine production. But in more complex organisms like humans, the presence of non-coding DNA and of regulatory genes means that the knowledge of the genome can't be easily translated into the proteome. The last thing you need to be aware of is that sequencing methods are continuously being improved, updated, and automated. This has increased the speed of sequencing and allowed the whole genome to be sequenced through methods such as high throughput sequencing. So the principles behind DNA sequencing, first of all, these techniques have become faster and automated compared to the original Sanger method, which was originally used, but many of the principles remain the same. The process involves terminator bases, which stop DNA synthesis, which is shown here in these coloured letters. Terminator bases A, C, G, and T are used. If an A terminator base is used, this will bind where a normal adenine base would have, and instead it terminates any further synthesis of DNA. So the basic principle is, firstly, DNA polymerase, a primer, an excess of nucleotides and terminator bases and the DNA to be sequenced are all mixed together. The mixture is in a thermocycler, which is used in PCR, which we'll come into soon, to synthesize DNA. The DNA polymerase will add bases complementary to the DNA, creating a new strand of DNA. The terminator bases are added at random and will terminate the sequence of DNA at a different point in each replicating strand. And that's what we can see here, so at different points in each replicating strand. This process continues until all of the possible DNA chains will be produced and the terminator bases at every single possible position have been made. So this is just a small sample here. Each terminator base, A, D, C and G, are labelled with a different fluorescent colour. That's why these are all shown in different colours. And that is then used to identify those bases. The DNA fragments are then separated using gel electrophoresis, and they separate according to their length. And now each fragment is arranged by length, and the terminator base is fluorescent to indicate which base it is. We can use that to work out the exact sequence of bases. So high throughput sequencing is when many fragments are processed and sequenced simultaneously, and this has made the process far more efficient. So once we had these genome sequences, we could make comparisons, and genome sequencing has made it possible for scientists to compare the entire genome of individuals of the same species and also of different species. So this can be used for analysing pathogens' genomes, and that has resulted in many advances, including identifying the source of an infection, for example MRSA, identifying antibiotic-resistant bacteria, tracking the spread of pathogens to monitor potential epidemics and pandemics, identifying regions in the genome for new drugs to target, and comparing genomes has improved the accuracy of classification of species, because we used to classify according to visible characteristics, but now we can classify based on how similar DNA-based sequences are for particular genes. It's also helped to understand our evolutionary relationships, because more similar the sequence of DNA bases in one organism to another will give an indication of how recently they've evolved from a common ancestor. Knowing the genome of an organism should mean that you can predict all the proteins that the organism can produce, but the human genome, as we've said a few times, it contains 20 to 25,000 genes, but the predictions for how many proteins humans can make vary from 17,000 to 1 million, and a lot more work is needed to be done on this to get an accurate estimate. But what this has taught us is that the relationship between the genotype and the phenotype is far more complex than we originally thought. So next time we look at synthetic biology, and sequencing genomes has enabled the development of synthetic biology, which means the creation of artificial pathways, organisms, devices, or the redesign of natural systems. Examples of this include genetic engineering, where we were able to isolate the gene for human insulin and insert it into a plasmid in bacteria so they can produce insulin. The use of biological systems in industry, such as immobilized enzymes, and the synthesis of new genes to replace faulty versions of genes. It's also used in the synthesis of new organisms. So, for example, new bacterial genomes have been created by scientists. So we then look at bioinformatics and computational biology. Computers are used throughout synthetic biology, including using bioinformatics, and that is the use of software to analyse, organise and store biological data. And this includes databases storing all the known alleles, amino acid sequences of proteins and structures of proteins. You also have computational biology, and that's the use of computers to study biology. So, for example, simulations and models and algorithms. And the protein structure can be modelled, including the effects of new mutations in the base sequence, and therefore the altered protein structure can be observed and compared to the functioning protein. Now, just to point out, I have added this in because in OCR it's often known as a short tandem repeat, STRs. It's AQA that use the term VNTR. So for OCR, you're more likely to see STR. If we have a look at VNTRs next, 95% of human DNA is made up of introns, and these are the sections within genes DNA sequences that don't code for amino acids. And they consist of variable number tandem repeats. So we have a variable number of bases that are repeating over and over. And the probability of two individuals having the same VNTRs is very, very low. However, the more closely related you are, the more similar your VNTRs are. So we've been able to analyse VNTRs through genetic fingerprinting to determine how closely related different individuals are. So the process of this genetic fingerprinting is, first of all, you have to collect your sample. And you can collect your sample from any cell that will contain a nucleus with DNA in. So this could be a blood cell, not a red blood cell though, it would have to be a white blood cell, different body cells, hair follicles. And if the sample of DNA is quite small, you would then put your DNA into a PCR machine to get lots of copies. So you've got a bank of that DNA so you can repeat this multiple times. Once you've collected and extracted your DNA, we then have to cut it up. And that's what the digestion stage is. And to cut it into smaller pieces, we use restriction endonucleases, and they cut the DNA at specific recognition sites, which will be close to the target VNTRs. We then separate out all of those cut up pieces of DNA using gel electrophoresis. So you load your different DNA samples from different individuals into small wells in agar gel. The gel is placed in a buffer liquid with an electrical voltage applied. And because the DNA has a negative charge, and at the other end of the gel there'll be a positive charge, the DNA will move through the gel towards the positive. And because some of those VNTRs will be shorter than others, the shorter ones can move faster, the longer ones move slower. That is what is going on in this separation stage here. Once we've separated those VNTRs, an alkaline is then added so that the DNA sections are split into single strands. It will break the hydrogen bonds and we now have single strands. The reason we need to have that is so we can hybridize your single strands with DNA probes, which are short, single-stranded pieces of DNA complementary to the different VNTRs. So the DNA probes will align opposite and hybridize, meaning forming hydrogen bonds, with those sequences. And this is needed so that we can see where those bands are, because the DNA itself isn't actually visible. So the DNA probes either have a radioactive label or a fluorescent label on them. Once you've allowed time for the probes to bind, you then rinse your gel, and we could then transfer all of that from the gel onto a nylon sheet, because when the gel dries it cracks and therefore you wouldn't be able to visualize your results properly. And the nylon sheet can then be exposed to X-rays to be able to visualize radioactive labels on a gene probe. Or if you use the fluorescent probe, you would use UV light to visualize the positions. And what you get is something a bit like this. And this takes me back a long time ago now, back in 2015, where I did this at Imperial College on a school trip with some of my students. And we had different samples of bacterial DNA. We had 5 species and an unknown. And we can see all of the different positions that the VNTRs moved to. We had a radioactive label and then we put it under an X-ray machine, and we can see here that the bands of the unknown are in the same position of the bands for species 3, and therefore we knew that our unknown bacterial species was bacterial species.
Electrophoresis can also be used on proteins, and this is known as protein electrophoresis. This is used in the diagnosis of medical conditions where an abnormal protein caused by a mutation in the DNA is responsible for the disease, so for example in sickle cell anemia. The method is very similar to that for DNA with 2 differences. As proteins have a complex 3D shape, they need to be denatured first so they can pass through the gel, and this is done by heating the proteins. As proteins can have positive, negative or no charge, all proteins need to be made to have a negative charge so that they move through the gel, and this is done by using a chemical called sodium didecyl sulfate, or SDS.
3. Next we move on to PCR, and this is going to be used at the start of basically all gene technologies because it creates a large sample of DNA so you can do multiple repeats and lots of experiments. And your equipment list for PCR would be this thermocycler machine. You need the DNA fragment you want to amplify. You need an enzyme, DNA polymerase, primers, which are short sequences of single-stranded DNA, which are needed to initiate DNA replication, and then DNA nucleotides. Now, the reason I've written Taq polymerase here is because it's a special type of DNA polymerase which is taken from bacteria that naturally grow in hot springs. So they have adapted to survive at very high temperatures, and therefore their DNA polymerase doesn't denature until very high temperatures. So that means we can do this reaction at very high heat without the enzyme denaturing. So here is our method of PCR. Step 1, we've added all of those ingredients and the temperature is increased to 95 degrees C. That will break the hydrogen bonds, so we now have our DNA sample as 2 separate strands. The primer will then align opposite complementary bases to that strand, and the primer has to bind to enable the DNA polymerase to be able to attach and therefore add all of the nucleotides. So for this to happen, we have to drop the temperature to 55 degrees C, so those hydrogen bonds will form between the primer. Then the enzyme DNA polymerase will attach all of the complementary DNA nucleotides and make the new strand. So that is our synthesis stage. And at that point, the temperature is increased to 72 to speed up the process. And it's also the optimum for the enzyme DNA polymerase. So the advantages of PCR is it's automated. You put all of those ingredients into your thermocycler and the reaction automatically happens. It's also very rapid. You could get hundreds of billions of copies of DNA within a matter of hours. And it doesn't require living cells like in vivo cloning does. And that leads us into what in vivo cloning is. And this is just showing you an overall summary of recombinant DNA technologies. So we've gone through genetic fingerprinting and gel electrophoresis and how that can be used to look at how closely related different organisms are. We've just gone through how PCR is a type of cloning DNA in vitro. So now we're going to have a look at how you can create DNA fragments and use those in vivo cloning. So we're going to start by looking at, once you've got your DNA fragment that you want to clone, we're going to see how you can insert it into the vector, into the host, which is the organism that will transcribe that DNA.
Then we look at restriction endonucleases, and these cut at recognition sites, leaving sticky. Restriction endonucleases are enzymes that cut up DNA, and they're also known as restriction enzymes. They actually naturally occur in bacteria as a defense mechanism, and there are many different restriction enzymes which have a unique shape active site to be able to bind to a particular complementary DNA base sequence. And therefore you have lots of different restriction endonucleases that can bind to lots of different DNA base sequences. And those particular DNA base sequences that they can bind to with the active site is called a recognition sequence. Each enzyme cuts up the DNA at a particular location. Some enzymes cut at the same location in the double strand, and that would mean it would cut directly down and create a blunt end. And by that we mean there's no overhang. Whilst others cut to create this staggered end on both sides, and that is these exposed bases. The exposed staggered ends are palindromic, which means it's the same forwards as it is backwards. And because of that, they're able to easily bind to complementary base pairs and stick back together. And that's why we call these sticky ends.
Once we've done that, we then need to insert this DNA fragment into our vector, which, as we said, is something to carry the isolated DNA fragment into our host, which is the bacteria. So plasmids are the most common vectors used. And plasmids are just circular loops of DNA, which are found in some bacteria. They're separate from the bacterial genome, which contains all of the genes. And plasmids typically only contain a few genes, and usually genes for antibiotic resistance.
Some extra information added for the 2025 version, and that is some extra vectors just here.
So the way we get our gene fragment into a plasmid is we cut it open using the same restriction endonuclease that was used to cut your DNA fragment. That creates sticky ends in the plasmid that will be complementary to the sticky ends on your gene fragment. Therefore, we can mix your gene fragment and the open plasmid together, and we'd add the enzyme DNA ligase to then join together your plasmid and your fragment at those sticky ends. So that is then going to enable the condensation reaction to form the phosphodiester bonds between the nucleotides to create this recombinant plasmid, meaning it's recombined, it contains DNA from two different organisms. Now we've got our vector that contains the gene of interest, we need to get that vector into the host cell, and that's what transformation means. So to do this, to get our plasmid into a bacterium, we need to make the cell membrane more permeable, so it's more likely for the plasmid to enter. So to increase the permeability of the cell membrane, the host cells are mixed with calcium ions and they undergo heat shock, which is a sudden increase in temperature.
Another method that you need to know about is electrophoresis, which is when an electrical current is applied to the membrane to make it more porous. And all of those then enable the vector to enter the host cell's cytoplasm, or at least make it more likely that it is able to.
So we should then get the recombinant plasmids entering. That won't always happen though, so we can see here we've got some bacteria that don't get transformed, and some bacteria do get transformed, but they take up plasmids that didn't contain the gene of interest. And that takes us on to this last idea of how you can identify which cells are transformed. And we use gene markers for this. So the three issues that might occur are, number one, the recombinant plasmid doesn't get inside of your bacterium, so it isn't transformed. Number two, maybe it does take up the plasmid, but the plasmid rejoined before it took in the gene of interest. Or number three, sometimes your DNA fragment actually just loops around and makes its own mini plasmid. So we need to check that the bacteria are bacteria that contain the transformed plasmid before you then spend time and money growing that bacterium. So marker genes are used for this, and plasmids contain marker genes. They normally have antibiotic resistant genes within them. There are different examples of what you could use, but you don't need to know the details of all of these. So we're just going to go through one example. So in this example, I'm going to talk you through antibiotic resistant marker genes, where we have a plasmid from a bacterium that contains a gene for resistance to ampicillin and a gene for resistance to tetracycline. And this is the DNA fragment that we want to insert into our plasmid. Now that will be deliberately inserted in the middle of one of these antibiotic resistant genes to disrupt it and stop that gene from working. So that means our recombinant plasmid will be able to produce the proteins that make it resistant to ampicillin, but it won't be able to make the proteins that make it resistant to tetracycline. So any bacteria that take up this plasmid will be resistant to ampicillin, but not tetracycline. So the way that we can identify them is following this method here. We'd grow all of the bacteria on an agar plate. We would then use a sterile block to take an imprint of these colonies and print them onto a new petri dish which has agar with ampicillin dissolved within it. And we can see which of these colonies were able to grow. And we can see A, D, E, G and I grew. What that tells you is C, H, B and F, they didn't grow. So that means they must not have taken up a plasmid at all because they died when they were grown in ampicillin, so they didn't have this ampicillin resistant gene on the plasmid. We would then take an imprint of these colonies and print it onto a plate which now just contains tetracycline. And this time A, D and I grow. So what that tells us is colonies E and G, they were resistant to ampicillin, but they weren't resistant to tetracycline. So E and G must contain that recombinant plasmid, so we could then take E and G and grow those en masse, and that would then clone our gene of interest inside of the bacterium. So that's an example of genetic engineering, and if we now have a look at genetic engineering in plants, the DNA of crops have had genes added to them to make them pest resistant, disease resistant and herbicide resistant, and all of those result in higher yields. Soy plants, for example, are a major crop across the world, and at least half are genetically engineered strains. One modification was the addition of a gene so they produce Bt toxin, which is toxic to pests. And that means that farmers don't have to add pesticides to kill the pests because they naturally have something within them which will do the killing. And that results in much higher yields of soya plants. Crops have also had DNA manipulated to have a longer shelf life to reduce food waste and also to increase nutritional value or even to produce medicines. So that all sounds great, but there are some perceived negatives, and that is that the genes for resistance to pests, disease and herbicides could spread to other plants in the environment. There are also concerns that people may be allergic to the different proteins that some of the crops might make. And one big concern is that the technology is often patented, and therefore buying genetically engineered seeds is very expensive and unaffordable for some farmers. Genetic engineering in animals is not as widely used and it's not as easy. Modified viruses and gold-covered DNA have been injected into animals to carry new genes into their DNA. This has been used to create swine flu-resistant pigs and faster-growing salmon.
For the 2025 edition, just to make you aware that I've added in some extra details here on farming, including these examples, so combining genetic engineering with pharmacology. So you might want to just double check you've got that written into your notes as well. In genetic engineering in microorganisms, farming is one of the biggest uses, and this is when a human gene is inserted into bacteria so that they produce a human protein, and a really common example is insulin producing bacteria. Microorganisms have also been genetically modified for research purposes and to act as vectors, so for example making non-virulent viruses for gene therapy. Concerns include increasing antibiotic resistance in bacteria, increased risk of cancer in patients receiving the viral vectors due to the fact that DNA is being inserted, which could cause mutations and possible disruption to the expression or regulation of genes by that inserted DNA.
Gene therapy is when human DNA is altered to treat disorders. So, for example, cystic fibrosis, which is caused by a recessive allele and results in increased mucus production in the lungs, amongst a whole range of other serious symptoms. The symptoms of excess mucus in the lungs can result in lung infection and shortness of breath, but it could be reduced with gene therapy. So the cells lining the lungs could have the defective recessive allele replaced or supplemented with a healthy dominant version of that allele, dominant version of that gene. In order for this to work, cells from the patients would have to be isolated. Then a viral allele, which has the desired allele inserted into the viral DNA, would be used to insert the allele of choice into the DNA of the patient from the isolated human cells. These genetically engineered cells would be injected or inhaled into the patient. These genetically engineered cells will then produce the desired protein, and this treatment is not a cure as it doesn't replace all the cells in the body with the faulty gene, it just supplements them. And this is an example of somatic gene therapy, as it's replacing body cells. Germline gene therapy is when you alter the DNA in a gamete so that the offspring would not inherit the faulty allele.
So if we compare somatic cell versus germ cell gene therapy next, the main differences are somatic cell therapy is often temporary and it needs repeating, whereas germ cell therapy is permanent. Somatic cell therapy only affects certain cells, whereas germ cell therapy would affect every cell. Somatic cell therapy only affects the individual, whereas germ cell therapy would affect the offspring of the individual as well. And that's because if you're editing the original germ cells that were used to create the new human or whichever organism it is, all of their cells will therefore contain the edited DNA, because once you have that sperm and egg fuse, the zygote then divides by mitosis, so they're all cloned cells and have exactly the same DNA in them. It also means that any sperm or egg cells that they produce will contain that edited DNA as well, which is why it's passed on to the offspring.
We then move on to cloning and biotechnology, starting with natural clones in plants. Now, many plants reproduce sexually and asexually, And due to their natural asexual reproduction, there are parts of plants that replicate by mitosis from stem cells, and that's known as a meristem. Strawberries form runners, which are horizontal stems that branch away from the plant and then form roots to reproduce asexually to form a clone. In some plants, the horizontal stems grow underground, and this is called a rhizome. This has been made use of in horticulture, as it means new plants can be created by free and quickly, as you're not waiting for a germinating seed, and farmers can also clone plants that have desirable characteristics. Another simple method is taking plant cuttings, and that's what we can see here. Plant cuttings involve cutting off a non-flowering stem from the budding plant, dipping it in plant hormones and also fungicide to prevent infection, and then you'd grow them in a soil. Micropropagation is a similar method that involves taking only a tissue culture, sterilising the sample, for example with ethanol, and growing with hormones such as auxin and cytokines on agar first until the roots are developed. A callus forms, and this is split into cells and transferred to a new agar plate, and eventually those small plantlets that form are potted in soil, and that's what we can see here. So we've got a summary here of the advantages and disadvantages of this micropropagation. So pause at this point and turn those into flashcards. Next, we look at natural clones in animals. And some invertebrates naturally clone themselves. For example, starfish. An entire new starfish can regenerate from fragments of the original starfish. In humans and other animals, identical twins are also natural clones, meaning monozygotic. And that is when one sperm and egg fuse to make a zygote, but that cell then splits into two to make two new embryos originating from that first zygote, so they're genetically identical. You can also have artificial cloning in animals, and the knowledge of how identical twins naturally occur has been used to produce identical animals in farming to maintain desirable characteristics. And this is known as embryo splitting, or essentially artificially making identical twins. So two parents with desired characteristics will be bred together. The female will be given hormones to make her produce many, many eggs. Samples of their eggs and sperm are used in IVF in the lab, or sometimes the eggs are naturally fertilised with the sperm. When an egg is fertilised and the embryo forms, which is just a small ball of cells, the embryo is then split apart into many single identical cells. And those identical cells are then inserted into the uterus of different host mothers, and all of the offsprings would be clones of each other. They wouldn't be identical to the female that they were grown in. They'd be identical to each other, but not their parents, because the parents had an egg and a sperm fuse. So they're not identical to the parents, they're just identical to each other. The second method used is somatic cell nuclear transfer, or SCNT. A somatic cell, which is a body cell, is taken from the animal that you wish to clone. An egg cell is taken from a female and the nucleus is removed. So here we have that body cell, the nucleus is removed that you want to clone, and you take an egg cell from a female and you take out the nucleus. Then what you would do is fuse together the nucleus of the animal you want to clone, and you fuse it into the egg cell. And to make it start to divide, a small electrical current is applied, and it will then start to divide to form an embryo. Once the embryo is a bundle of cells, the cells are separated and inserted into the uterus of different host mothers. Each of these inserted cells is genetically identical to the parents that nucleus was taken from, but also to each other. And this is how Dolly the sheep was made. Dolly the sheep was the first animal to be cloned using SCNT. She was healthy, but had a much shorter life. It's not known why animals produced by SCNT have a shorter lifespan, but it's thought to be linked to the fact that they are created from older adult DNA, which has already aged. So here we have the pros and cons of this animal cloning, which again you can turn into a flashcard, or 2 flashcards here for the pros and 1 for the cons.
Now this is an important 2025 edit that I want to make you aware of. You no longer need to know examples of microorganisms in biotechnology, but it is still helpful to be aware of them. So over the next few slides, you don't need to know this information. However, it's useful to have an understanding of it and know examples.
Then we move on to microorganisms in biotechnology. And as the human population continues to increase, the demand for food increases at a rate that cannot be matched by the space and time for farming animals. Therefore, microorganisms have been used to try to solve this. So, for example, bacteria and fungi have been used, and that is because of economical advantages. So microbes are tiny, so you can grow a lot of them in a small space compared to animals. Their nutrient requirement is also cheaper than animals, and the temperatures that they grow at are low, which makes harvesting them cheaper. They also have a short life cycle, so the food production is much faster than it would be for growing animals. Their growth requirements are very basic, so they're easier to grow. And microorganisms have been used in food production to create the following. So in breads, yeast is a fungus used in baking, and when the yeast respire, they produce carbon dioxide, which helps to make the bread rise. In brewing, when yeast respire anaerobically, they produce ethanol and carbon dioxide, and the ethanol is used to make alcoholic drinks. Cheese use bacteria, and bacteria will use lactose in milk as a respiratory substrate. And this causes the milk to start to separate into the solid curds and the liquid whey. And those solid curds are then used to make cheese. Yogurt also involves bacteria, so species such as Lactobacillus bulgaricus and Streptococcus thermophilus, and that's because they produce ethanol or lactic acid, which results in the milk forming these polymers that give yogurt a thicker and smoother texture compared to milk. Mycoprotein, or the brand name Quorn, is produced from the fungus Fusarium venetatum, and it's grown and used to make proteins that make up corn. Other examples involve the use of microorganisms in medicine. So penicillin and insulin are both medicines produced by microorganisms. Penicillin is the antibiotic produced by a particular fungus, and insulin is produced by genetically modified bacteria, as we just went through. Bioremediation is when microorganisms are used to digest pollutants and contaminants. So naturally occurring microbes can break down pollutants like sewage and crude oil, and microbial growth can be encouraged by giving them the nutrients they need to grow. This technique is common on contamination sites, such as we can see here, if there has been an oil spill. So then we come to our evaluation of the use of microorganisms in biotechnology. So pause and have a go at turning that into a set of flashcards. Then we have a look at how we can culture microorganisms. So when they're cultured, you have to have a large enough quantity to see without a microscope. And microbes must be grown with a growth medium, which could be a liquid broth or solid nutrient agar. And that would contain water and nutrients such as glucose and amino acids. They also require oxygen, and in schools they're incubated at 25 degrees C because that's warm enough for growth, but not so warm that you would potentially have large quantities being produced, which might then be pathogenic and cause harm. When you're growing microbes, you also have to work aseptically, which means in complete sterile conditions, to make sure that you don't have contaminations of the bacteria you're growing, and so you don't infect others. So this includes sterilising all equipment. So you could use an autoclave, or for metal equipment, you might put it into a roaring flame on the Bunsen burner until it glows red. You have to sterilise all the work surfaces with disinfectant, wash your hands with soap, and you always work near a Bunsen burner because there'll be a convection current that continually sterilises the air near the Bunsen burner, so you get this sterile dome of air that you could work in. You should only open the Petri lid. You should only open the lid of the Petri dish slightly to again reduce the chance of microbes within the air landing on your dish and contaminating your sample. And lastly, as we said, sterilise all the equipment, which could be flaming it in a Bunsen burner.
So if we have a look then at batch fermentation, in this type of fermentation, all of the necessary nutrients are added at the beginning of the fermentation process. and the culture is allowed to grow until the desired product is obtained or until growth is limited due to the depletion of nutrients or the accumulation of toxic inhibitory products. Manipulating growing conditions such as temperature, pH, oxygen availability, agitation rate and nutrient concentrations can significantly impact microbial growth, metabolism and product formation. For example, controlling the pH within an optimal range can ensure maximum enzyme activity or product yield. Similarly, maintaining the optimum temperature and oxygen levels can promote cell growth and enhance product formation. Sampling and analysing the culture during different stages of fermentation allows for real-time monitoring of that microbial growth and product formation, and it enables you to adjust any of those conditions if you need to make sure you are achieving maximum yield. Now, the other type of fermentation is continuous fermentation, and this involves a continuous flow of fresh, sterile medium into the bioreactor while simultaneously removing an equal volume of the spent medium containing the product and cells. Manipulating growing conditions in continuous fermentation is essential for achieving steady state conditions and maximizing productivity. So these conditions include the dilution rate, the nutrient concentration, residence time and bioreactor configuration, and those are all needed to be looked at and controlled to determine first of all the product yield and quality in the continuous fermentation. You do also need to monitor and control other factors such as pH, temperature, dissolved oxygen levels, because those are all going to have an impact on enzyme activity and also respiration, and therefore the microbial activity and metabolic efficiency.
So then we look at the standard growth curve of microorganisms. Bacteria reproduce rapidly, approximately dividing every 20 minutes. Therefore, you can get large quantities very, very quickly. And when looking at the population size when culturing bacteria, log scales are often used for that reason, because you have such a large range in the number of values. To work out the number of individual organisms, you can use the following formula. N equals N0 times 2 to the N, where N0 is the initial number of bacteria, and N is the number of divisions or generations. When the log of the numbers of bacteria grown over time is plotted on a graph just like this, it's called a growth curve. And we have these 4 key stages: the lag phase, exponential phase, stationary phase, and the death phase. So in that lag phase, the number of bacteria is lower while the bacteria are adjusting to the new environment. Once they're reproducing at their maximum rate, the increase in the number is exponential, and that's how we get this exponential phase. They will reach a point where the number of bacteria from growth is equal to the number that dies, and that is where we get this stationary phase. The death phase is when the number of bacteria decreases because the number dying now exceeds the number being reproduced. And the reason they start dying could be they've run out of resources such as glucose, or it could be toxins are being built up and that starts to cause the death of them. The death phase could theoretically be prevented, and you go through this stage, if you remove the limiting factors. So making sure they have plenty of nutrients, there's plenty of oxygen, maintaining the temperature and removing the toxins, and also buffering the pH. An alternative to using microorganisms is to isolate the enzymes they contain and then immobilize those enzymes. And this is an advantage as it's even more efficient, more specific, and you don't even need growth mediums because it's just the enzymes. So immobilized enzymes are when the enzyme required is fixed to an inert substance and the substrate is then passed over it. This is beneficial as you do not need to purify the enzymes from the product at the end, as the enzyme remains attached to that inert object. That means you can easily reuse them and save money, and having the enzyme fixed also decreases their sensitivity to temperature, so you can use higher temperatures without the enzyme denaturing. This method does have upfront costs, and it is more technical. So here we have our pros and cons, which you can summarise on flashcards. The examples of the use of immobilised enzymes include glucose isomerase for the conversion of glucose to fructose, penicillin and acrylase for the formation of semi-synthetic penicillins, lactase for the hydrolysis of lactose to glucose and galactose, aminocyclase for the production of pure samples of L amino acids, and glycoamylase for the conversion of dextrins to glucose.
An important little 2025 notification here is you are not required to recall the examples above, but you will be expected to apply your knowledge and understanding of immobilized enzymes in the context of biotechnology. So you could come across these in the exam, but you don't have to remember those examples.
Next, we move on to ecosystems. And we start with lots of key terms that you need to know the definitions of. So pause at this point and turn these into flashcards and learn these key terms. So ecosystems and biomass. Ecosystems are dynamic, meaning they are constantly changing, and they vary in size, which is determined by the biotic, living factors, and the abiotic, non-living factors within the ecosystem. So for example, a rock pool, a playing field, and a large tree are all very different sized ecosystems. Biomass gets transferred through ecosystems, and in any ecosystem, plants are the producers of a food web, and they are able to produce their own carbohydrates using carbon dioxide in the atmosphere or from carbon dioxide dissolved in water. Between each trophic level in a food web, the majority of energy is lost due to respiration and excretion, and the remaining energy is used to form biomass. And biomass is the dry mass of the carbon-containing compounds. So the given amount of biomass remaining in an organism can be measured in terms of the mass of carbon or dry mass of tissue per given area. How productive an ecosystem is depends on the abiotic and biotic factors. So if there's plenty of water, light, warmth and green plants, that will maximise the rate of photosynthesis and therefore a more productive ecosystem because more carbohydrates will be produced. The efficiency of biomass transfers between trophic levels can be calculated using this formula. So the biomass transferred divided by the biomass taken in times 100. And humans can manipulate the transfer of biomass through ecosystems by reducing energy lost in trophic levels, and this is done often in farming. So you could restrict the movement of your animals, you could provide the animals with higher energy food, you could keep them indoor to reduce heat loss, and by removing competition and predators, they'll also be expending less energy running to compete or running to evade predators. We then move on to the nitrogen cycle, and the air is 78% nitrogen. However, plants and animals can't obtain nitrogen through gas exchange, and that is because nitrogen has this triple bond between nitrogen atoms. So microorganisms are needed to convert nitrogen gas into nitrogen-containing substances that plants and animals can absorb. So the reason nitrogen is so important is it's found in proteins, ATP and nucleic acids, which are all essential for survival. The nitrogen cycle is split into 4 different key stages, and you also need to know the importance of saprobiotic nutrition in organisms and also microbes in general in this process. So here is our nitrogen cycle, which we'll talk through. Let's start with nitrogen gas. That nitrogen gas can be converted into nitrogen containing compounds such as ammonium through bacteria, which can break the triple bond. And that is nitrogen fixing bacteria, which is naturally found in the soil, or nitrogen fixing bacteria that naturally occurs in the root nodules of leguminous plants. So if we carry on with the nitrogen fixing bacteria in the soil, they're able to convert the nitrogen gas within the air in the soil into ammonium. That ammonium is then oxidized by nitrifying bacteria into nitrites, and we call that stage nitrification. The nitrites are oxidized further into nitrates, again by nitrifying bacteria. And those nitrates can then be absorbed into the plants by active transport, and then the plant will use those nitrates to make proteins, DNA, and ATP. Those plants will then be eaten by animals, and the protein in the food that they're eating will be digested to release those nitrogen-containing compounds, which can be assimilated into the proteins and DNA, RNA, and ATP for the animals. Now, animals will produce waste, and both animals and plants die. And when waste is made or when organisms die, this is where we have microbes involved yet again. And this is our saprobiotic nutrition, or the saprobiotic microbes, because they are able to release enzymes that will digest the proteins in the dead plant matter or dead animal matter, or to digest and break down the urea in excretion into ammonium. And in that way, we then loop back round to the cycle. Now, there's one branch here that we haven't talked about, and that is when nitrates could be converted back into nitrogen gas by denitrifying bacteria. But this only happens in anaerobic conditions. So if the soil becomes waterlogged or too compact, and therefore there's not enough oxygen in the soil, the denitrifying bacteria thrive in anaerobic conditions, and therefore they will be able to convert that nitrate into nitrogen gas. And that is why in agriculture, they'll often plough and toss the soil around to make sure it remains aerated. So this just shows you lots of different bacteria are involved and they're important in the nitrogen cycle.
So that was the cycle, but let's just go through the exact names of those bacteria, as you do need to know those. Nitrogen fixing bacteria, you need to know about Rhizobium, Azotobacter, and these are the ones that can break the triple bond between those two nitrogen atoms when it's as nitrogen gas in the atmosphere. The bacteria are either free living, which is the Azotobacter, or it could be the symbiotic, mutualistic ones that live in the root nodules of leguminous plants, such as, or that is, the Rhizobium. In nitrification, we said that we have nitrifying bacteria. We have the nitrosomonas, which convert ammonium to nitrite, and then the nitrobacter convert nitrite to nitrates. Then we have denitrification, and this stage is not useful, as we said, and it returns the nitrogen in compounds back to nitrogen gas in the atmosphere, so it cannot be absorbed by the plants. That is by anaerobic denitrifying bacteria. And then lastly, the ammonification stage, that's when proteins, urea and DNA are decomposed in dead matter and waste by saprobionts. And those will be organisms such as bacteria and fungi, which digest waste extracellularly and then return ammonium ions to the soil.
We then move on to the carbon cycle, and the carbon cycle is also dependent on organisms. We start with plants, which can photosynthesize to fix carbon from the atmosphere in the form of carbon dioxide into carbohydrates. Those carbohydrates will then be eaten by animals that are ingesting and digesting the plant material. All organisms respire, and this will convert the carbon in the carbohydrates back into carbon dioxide in the atmosphere or the oceans. When organisms die or through excretion, the carbon is broken down to carbon dioxide by decomposers that respire using the carbohydrates within the dead or waste matter. An imbalance in this cycle is leading to the oceans becoming more acidic and also global warming. And this imbalance is caused by deforestation, which can result in the trees being burnt, releasing more carbon dioxide. But also, if you are cutting down trees, less carbon dioxide is being taken in by photosynthesis. We then move on to succession, and this is a change in an ecological community over time. A primary succession always starts with a pioneer species, which is the first species to colonize an area, which could be bare rock or sand. The pioneer species is often something like lichen, which is adapted to survive in harsh abiotic factors. And through their death and decomposition, that changes the abiotic factors to make them be less harsh. You often get this thin layer of humus forming, this soil layer, and that now means other species are able to colonize, and they will actually outcompete the pioneer species. So that is when we get our mosses and smaller plants that are now able to survive. As they die and decompose, that will add to the soil further and increase the nutrients. So the abiotic factors again become more and more favourable. And this pattern continues to occur, and the abiotic factors become less harsh and larger plants are able to survive. Each new species changes the environment in such a way that it becomes less suitable for the previous species. Therefore, each existing species is outcompeted by a new species colonising. Overall, during a succession, we increase the biodiversity, the abiotic factors become less harsh, and therefore we have a more stable environment. And we get to the final stage, which is known as the climax community, which is often dominated by large trees. So in summary, the species richness and the number of organisms increases. So in other words, your biodiversity increases. And as succession occurs, larger plant species and animals will be able to colonise the area. A deflected succession is when human activities can prevent the progress of succession. A climax community won't be reached if there are animals grazing or if there are humans that are trampling an area. Or you could have controlled burning and the removal of vegetation, both of which prevent a climax community forming. And this is your deflected succession. Now, the reason this might be done is to maintain the earlier stages in succession so that you get a greater variety of habitats being conserved and therefore a greater range of food sources and species being able to survive. This can lead to a conflict between human needs and conservation in order to maintain the sustainability of natural resources. So to try and manage this conflict, compromises are needed. For example, forests can be coppiced to provide timber for fuel and furniture, while still allowing a tree to survive. Now, we can estimate population sizes through sampling. And the reason we do sampling is it'd be too time consuming and inaccurate to say you were going to count every individual within a population in an area. So that's why we use sampling. And population sizes can be estimated using various techniques, but a few aspects are commonly used. For sampling to provide an accurate estimate that is representative of the population size, many samples, and that means 30 plus ideally, should be taken, and it should be random sampling to avoid bias. Lastly, we move on to populations and sustainability. So we start by looking at factors that affect population size. And we come to abiotic factors, which we have touched on briefly already. So these are the non-living factors or the environment. And that could be temperature, oxygen, carbon dioxide, and all the others listed there. Plants and animals are adapted to the abiotic factors within their ecosystem. And these adaptations develop through the process of natural selection over time. The less harsh the abiotic factors, such as plenty of water and light, the larger the range of species and the larger the population sizes. Biotic factors are the living factors in an ecosystem, and this can include interspecific and intraspecific competition and predation, and both of those will affect the population size. Interspecific competition is when members of different species are competing for the same resources, whereas intraspecific competition is when members of the same species are in competition for the same resources and also a mate. And competition for a mate links to courtship rituals, because individuals that are fitter will have more energy to perform a more impressive courtship ritual, or they may have more fur or feathers in a better condition to attract a mate. The idea of predation links to these predator-prey graphs, and regardless of the species, the graph always follows the same pattern. The size of the predator and prey population will both fluctuate. There is typically more prey than there are predators because they are the food source for the predators, so there has to be more of them to sustain the predator population. And lastly, the size of the prey population will typically change before the predator population. And that's what we can see here. The prey starts to decrease because they're being eaten by the predators, but because they are now decreasing, we then get a decrease in the predators. Because the predators have decreased, the prey can then start to survive and we get an increase, but then that results in more food for the predators, so they increase. And that pattern continues over and over.
2025 edit here to make it really more scheme specific is just to be aware that conservation in human actions and management is to help increase biodiversity. And you would have to say increase to get the mark, not maintain.
Then move on to conservation and preservation. Conservation is human actions and management to help maintain biodiversity. And it involves sustainable development, whereby management of ecosystems is in place so that natural resources can be used by humans without them running out. Preservation involves protecting an area by banning visitors. And this is a very effective method of protecting ecosystems, but it does prevent anyone from enjoying the area. Conservation is really important for three reasons. The ethical reasons, so all organisms have the right to live, and conservation helps to ensure humans are not preventing this. It also helps ensure future generations of humans are able to experience and enjoy those natural ecosystems. There are the social reasons, which include enjoying the outdoors and providing many physical and mental health benefits to people. Then there's the economic benefits. Many medicines and foods and clothes and even timber are sourced from different ecosystems, and also it can be involved in tourism. As the human population continues to increase, sustainable management is required to ensure that humans have food, shelter and infrastructure for survival, but without causing excess loss of biodiversity and the depletion of resources. And that is what sustainable management is. For example, sustainable timber production involves coppicing and pollarding. And these techniques are when the trees are cut close to the ground to be able to provide timber, but the plant will still regrow. On a larger scale, when larger forests are felled, only the largest trees are removed and new trees are then replanted to replace them. Sustainable fishing is essential to protect future fish populations while still providing a valuable protein-rich food source. There are policies in place which include limiting the quantity of fish that can be caught, the size of the fish as well, the size of the holes and the nets that are used, and having certain areas that aren't allowed to be fished in, particularly during mating season.
So just to make you aware, on this next slide coming up, this is a 2025 change to the spec. You no longer need to know these examples, but it does state you need to have an awareness of the impact. So it's still useful to have this awareness, but you wouldn't have to know these literal exact examples in the exam.
We then move on to the control of human activity, and there are certain examples that you need to know. For example, the Masai Mara region in Kenya. The Masai Mara National Reserve is an ecosystem under management to ensure the conservation while not negatively impacting the need of humans. For example, the tsetse fly is a carrier of African trypanosomiasis, and to reduce the spread, acacia bushes have been removed as this is the habitat of the tsetse fly. As local tribes are not allowed in the park, they now have to graze their livestock on the outskirts of the park instead of within it, and this has enabled vegetation within the park to recover. Ecotourism is popular here, which provides economic benefits, but tourism must not exploit the natural environment or local communities. The nature reserve also plays an important role in protecting endangered species such as the black rhino. So to help strike a balance, legal hunting involves the culling of overpopulated animal species only. Another example is the Terai region in Nepal. And along the southern border of Nepal is the Terai region, which has very fertile land and well-watered floodplains. And for that region, it attracts many people because you'll be able to grow lots of crops and food there. So management is needed to ensure that overpopulation doesn't harm the environment. This includes a sustainable forest management in this region to provide a livelihood for the locals whilst protecting the forest. To ensure agriculture can sustainably occur, farmers are encouraged to grow crops further up the hills, improve irrigation, grow more than one crop variety to increase biodiversity, and to grow leguminous plants because they fix nitrogen. The next example is peat bogs. And peat bogs are carbon sinks that are wet and contain decomposing plant matter. As it is so carbon rich, if it was to be dried out, you would get a good fuel source. However, in burning that, you would now release lots of carbon dioxide in the atmosphere that previously was contained within the peat bog as a carbon sink. Peat has also been used in compost to improve the soil structure and acidity. So to help protect these peat bogs and therefore prevent this excess release of carbon dioxide into the atmosphere. So that takes us to the end of Module 6. I hope you found it helpful. If you have, don't forget to click like and subscribe so you don't miss out on any of the latest videos.